Now, this section will help you understand the conduction system of the heart. Let's have a look. The conduction system of the heart is the network of nodes, cells, and signals that controls the heartbeat. Each time when heart beats, electrical signals travel through the heart. This signaling is coordinated by the cardiac conduction system. This system is also known as the nodal system, and it is mainly composed of sinoatrial node, or SA node, atrioventricular, or AV node, and some conducting cells that are mainly found in internodal pathways that distribute contractile stimuli to atrial muscle cells. These conducting cells include bundle of His, bundle branches, and Purkinje fibers. Now, let's see what is the function of this conduction system. We know that the heart has to pump the blood throughout the body, and this pumping action should be properly controlled so that it can provide blood to each and every cell of the body in timely manner. So, for the heart to pump efficiently and the systemic and pulmonary circulations to operate in synchrony, the events in the cardiac cycle must be coordinated. Here, conduction system plays its role. We already mentioned that the conduction system actually controls our heartbeat. Every time when heart beats, electrical signals travel through the heart. These signals cause different parts of the heart to expand and contract. This expansion and contraction control blood flow through the heart. The conducting system also provides the heart its automatic rhythmic beat. As depolarization and contraction of cardiac muscle are intrinsic and do not depend on the nervous system. That means the heart can continue to beat in rhythm even when all nerve connections are severed. However, a normal healthy heart has many autonomic nerve fibers that are able to alter its rhythm. Conduction of cardiac impulse. Some cardiac cells act as autorhythmic cells that continuously depolarize and move slowly towards threshold. And at that level, they initiate action potentials. Action potential is a sudden rapid change in resting membrane potential. This action potential or impulse that generates in conducting cardiac cells passes through the heart in the following order. Starting from sinoatrial or SA node. It is a region of specialized myocytes located in the right atrium, close to the opening of superior vena cava. Because it generates the heart's rhythmic contractions, it is often referred to as the pacemaker of the heart. The SA node cells can reach threshold on their own, initiating impulses through the myocardium, stimulating contraction of cardiac muscle fibers. The path of a cardiac impulse then travels from the SA node into the atrial syncytium. Due to this, the atria begin to contract almost simultaneously. This action potential is then channeled through the atrioventricular node, located between the right atrium and right ventricle. The impulse is then passed through the junctional fibers of AAV nodal pathway to a mass of specialized tissues called the atrioventricular node. It is located at the junction of right atrium and right ventricle. The AV node provides the conduction pathway between the atrial and ventricular syncytia. Here at this point, impulses are slightly delayed for 0.1 seconds because of the small diameter of the junctional fibers. The atria therefore have more time to contract and empty all their blood into the ventricles before ventricular contraction occurs. When the cardiac impulse reaches the distal AV node, it passes into a large AV bundle, or bundle of His, entering the upper part of the interventricular septum. The atria and ventricles are not connected by gap junctions even though they meet each other. The only electrical connection between them is the AV bundle. The AV bundle soon splits into the right and left bundle branches. These branches move along the interventricular septum toward the apex of the heart. Nearly halfway down the septum, the right and left bundle branches spread into enlarged Purkinje fibers, extending into the papillary muscles. The fibers consist of long strands of barrel-shaped cells with only a few myofibrils, which continue to the heart's apex, curving around the ventricles and passing over their lateral walls. They complete the pathway through the interventricular septum. Purkinje fiber stimulation causes the ventricular walls to contract in a twisting motion to force blood into the aorta and pulmonary trunk. From the time the SA node generates an impulse until depolarization of the final ventricular muscle cells takes only 220 milliseconds in a healthy heart. In the absence of any extrinsic stimuli, this conduction system maintains the rhythmicity of the heart, and the SA node pacing rate will be 100 beats per minute. But the heart rate must vary in response to the needs of the body's cells for oxygen and nutrients under different conditions. To respond quickly to these changing conditions, this self-conduction is not enough, and the heart or cardiac contractility also needs to be regulated by nervous system. Here in nervous system, the autonomic nervous system, that is the component of peripheral nervous system, controls the cardiac muscle contractility. The autonomic nervous system has both sympathetic and parasympathetic divisions that work together to maintain balance. Where sympathetic stimulation increases the heart rate, that is the positive chronotropic effect, and also increases the force of contraction, that is the positive ionotropic effect. Under some stress, emotional disturbance, or during exercise, etc. While parasympathetic stimulation decreases the heart rate, that is the negative chronotropic effect, and decreases the force of contraction, that is the negative ionotropic effect. During rest, sleep, or emotional tranquility, the parasympathetic system may predominate. The sympathetic input into the heart is via the postganglionic fibers from the sympathetic trunk, which innervate the sinoatrial or SA node and atrioventricular or AV node. The stimulated postganglionic fibers then release noradrenaline. On the other hand, the parasympathetic input into the heart is via the vagus nerve. The vagus nerve forms synapses with postganglionic cells in sinoatrial or SA node and atrioventricular or AV node. When stimulated, they release acetylcholine. That decreased the slope of the pacemaker potential, thus leading to a decrease in heart rate. So this was all about the autonomic control of the heart rate. Let's move to the next section.